If your name is Sophie from Mamma Mia and you want to know which one of these people is your real dad, what do? Well, besides doing all sorts of funny schemes, you could also do a DNA profile. A DNA profile is basically a genetic fingerprint that is unique to each person. One way that we can distinguish someone's DNA from another person's is to look at short tandem repeats. The DNA code is written with 4 nucleotides, A, T, G, and C. Short tandem repeats, or STRs, are repeating segments of non-coding DNA, which basically mean that they are repetitions of a nucleotide combination. Most everyone has short tandem repeats using the same combination of nucleotides in the DNA, but the lengths are different. However, DNA is passed on from parent to child, so if you're related, your DNA profile and your STRs are going to be more similar. So how do we obtain the DNA profile of these candidates? First, we've got to get a cell sample from each of them, and then extract the DNA from the cells. Next, the STRs have to be cut out of the long strand of DNA. To do this, a special type of enzyme called restriction enzymes, which can recognize the ends of the sequences, identify the STRs, and cut it. Now that we have everyone's STRs, we can start seeing how they're different. Remember that most people have the same STRs, but where they differ are in lengths. This is where gel electrophoresis comes in. Gel electrophoresis is a process that separates molecules, like DNA, according to molecular size. Since we are talking about DNA, the longer the sequence is, then the bigger the molecule. Let's see how it works. The setup for gel electrophoresis looks something like this. Basically, you have a slab of gel made of agarose with indentations at one end of the gel, called wells, where the DNA will go in. The gel is placed in a box called a gel box, connected to a power source, which when on will run an electric current through the gel. When you have an electric current, there's a negative end, which is placed near the wells, while the positive end is on the other side. Now we're going to place all the STR sequences from each person into each of these wells. When the power source is turned on, the DNA, which is negatively charged, is going to want to gravitate toward the positive end of the electric current because opposites attract. The pieces of STRs that are larger are going to travel slower, and smaller pieces are going to travel through the agarose gel much faster. After a certain amount of time, you're going to end up with something that looks like this. These are called bands of DNA. The closer to the wells the band is, the longer it is, and the further away, the shorter. This is how we can visualize the length of everyone's STRs. So how do we know who Sophie's father is? Well, from gel electrophoresis, we got the DNA profiles of these candidates, and we can do the same to Sophie. Now compare all of their DNA profiles, and whichever one is closest to Sophie's, that would be her father. According to a mock investigation, it looks like Harry's the father, so there you have it. DNA profiling using short tandem repeats is just one way of looking at someone's DNA. It's not just used in determining parentage, but also in other areas like forensics. This is just a little snippet into DNA profiling, but I hope you found that helpful.